another member of the Alveolata, which is the A in SAR, is the phylum Ciliophora. And Ciliophora means bearing cilia. Phora is like a carrier, like an amphora. Cilia are cilia. So these are known as the ciliates. So rather than moving by flagella, that's something we saw a lot in the excavata, they move by means of cilia. And what I'm going to draw for you is, as a representative of the ciliates, is a species of Paramecium. Paramecium multimicronucleatum. That's a mouthful. Kind of reminds me of like a spell right out of Harry Potter or something. Paramecium multimicronucleatum. Razzle-dazzle. Okay, so Paramecia are free-living, typically aquatic organisms. And we'll see something that we saw previously in Euglena. We're going to see that again. Okay, so we're going to start with sort of a bean shape here. And this structure here, what we're going to find in the ciliates is they're thought to be some of the most complex single-celled organisms that have been encountered. Because they have a lot of structures that are somewhat analogous to things we see in multicellular structures like animals. So starting with a structure that's analogous to having like a mouth. So they will have an oral groove, as it is called, leading into a cellular throat. And a cellular throat is not what it's called. It's called a cytopharynx. Cyto means cell. Pharynx means throat. So that's kind of how we get cell throat. Cytopharynx leading down to a cytostome or a cell mouth. I'll go ahead and write cell throat here. Okay, and this is where our kind of analogy to multicellular organisms like animals that eat food kind of breaks down. Because they don't have a digestive tract like a gut. But at the cytostome is where they form food vacuoles. The food vacuoles will fuse with lysosomes. So we can just kind of draw this food vacuole. And then once the good stuff has been digested, there's going to be some metabolic waste. And that's going to leave our ciliate through the cytoproct. And maybe you're thinking cytoproct, like the proctologist, the butt doctor, would be correct, would be accurate. Cytoproct is like the cell anus. Okay, and so you might say to yourself, well, we've got this single-cell organism. Doesn't the plasma membrane Why does this plasma membrane need to have all of these kind of specialized parts to it? And for that, we need to draw some of the external anatomy here. And yes, I am going to draw these little lines going all the way around. If you see one of these under the microscope, you're not going to be able to see all of these little hair-like projections. Hair-like projections. What am I possibly talking about? Are they cilia? Yes, they are. Remember, we're in the eukaryotes now, and eukaryotes do have cilia. Prokaryotes do not, because cilia are powered by microtubules, part of the cytoskeleton that we don't see in prokaryotes, but we do see in eukaryotes. So we do have ciliates that have cilia. And the cilia are embedded in a complex series of membranes. So it's not just like one plasma membrane and then the environment. There's actually several layers of the membrane. And they're so complex that they've been analogized to being like a little cellular skin. And that little cellular skin is called a pellicle. Pellicle literally means like little skin. So pel, if you studied French, you know that the word for skin in French is peau, P-E-A-U. In Spanish, it's piel, P-I-E-L. Both of us, those words come to us from the same Latin word root, pel. Icle means like a little thing, like a molecule is a little bit of stuff. A pellicle means basically it's like a tiny little skin. And that's actually something we see not just in the Ciliophora, but in all members of the Alveolata. So alveolata get their name from having these complex pellicles that have little kind of bubbles of membrane embedded in the pellicle that look like, are reminiscent of the alveoli that we have in our lungs that are like clusters of grapes. So these little kind of round kind of membrane cysts, you could call them. That's where we get the name alveolata, which is the synapomorphy of the A in SAR. Okay. So what else do we have? We also are going to have, the usual organelles. So I'm going to draw two circles, but one of them is going to end up with a nucleolus in it, and the other I'll label in a second here. Now we've got a nucleus, but if you are good at decoding here, multimicronucleatum means that we've got micronuclei and several of them. And we have one macronucleus. One big nucleus and several small nuclei. And why is it? Well, it has to do with the rather complex life cycle of ciliates. Everything about them is very complex for single-celled organisms. And I'm not going to get into how that all works, but it has to do with different levels of ploidy. So there's that. That was my dog sneezing. Miss Ilio. Silly girl. Okay, so what's this thing over here? Well, I'm going to draw, bless you. some of these structures around it that makes it look like, kind of look like it's got the sun living inside of there, but it's not the sun. Again, like we saw in Euglena, another freshwater single-celled organism, when you live in fresh water, your habitat is trying to dilute you as much as it can because of osmosis. So what we have here is, once again, a contractile vacuole that squeezes out the excess fresh water, which enables the cytoplasm to remain hypertonic with respect to the surrounding environment. Okay, let me see. I think I've got all of the pieces in here that I would like for you to know, and I might feel comfortable asking you about in the context of a summative assessment. That's an exam. So yeah, there is your Paramecium multimicronucleatum. And yes, all the other organelles are in there as well, but we don't need to draw those.